right, good morning everyone. Going to start this next session with case examples of the work we're doing in Friedreich ataxia and talking about the FAI-CD and RDCA-DAP efforts for this disease. I'm just going to set the stage with a few slides and then we go into a panel discussions with a Q&A. What's the big red? I want to see a green. A big green then. All right. So a few disclaimers that we're going to pretend to read for 5 seconds. There we are. Today, really I want to discuss the Friedreich ataxia databases that we have at CPAF, the FAI-CD, historically the first database for Friedreich at CPAF, and then the RDCA-DAP, discuss the role of data sharing and aggregation with our panelists, some of the solutions and impact for the disease that we are already having, including informing drug development for Friedreich ataxia. So just historically, and we have Jen Farmer here from the Friedreich Ataxia Research Alliance to discuss further this effort that really started a big database for Friedreich ataxia that we have at CPAF. So this effort is called the Friedreich Ataxia Integrated Clinical Database, or FAI-CD, that was started in 2018 with really an impetus to accelerate research and drug development for the disease, standardize multiple data sets that were already available at the time, including natural history studies and clinical trials, into CDISC, the regulatory grade standard for data, and also make that data publicly available through this database, internal database at CPAF, where approved researchers could access and download some of these patient-level data that we were able to aggregate over the years. That was really a work started upon FERA wanting to collaborate with CPAF, and it has been a collaboration ever since between CPAF and FERA, and then FERA and RDCA-DAP. So you can see here the entry portal that is still available on the CPAF website, where you can potentially request access to some of the data for Friedreich ataxia. This is the current database, so as you can see, it's pretty compelling. It's probably the biggest rare disease database that we have at CPAF after Duchenne muscular dystrophy. That includes a big natural history study conducted by FERA called the FA-COMS, multiple clinical trials and control arms for clinical trials from industry. Recently, that database is growing. So recently, EFACTS, the European Foundation for Friedreich ataxia, joined the FAI-CD team and also shared their data. It's a multi-site collection of data across European countries, which is under curation right now and will be available soon on RDCA-DAP. Additionally, FERA shared a new natural history study, this time for pediatric Friedreich ataxia, adding again to the database. There will also be an update of the FA-COMS natural history study that will come soon to add and increase the current data that we have for the FA-COMS study. Also recently, our team got access to an EHR synthetic data set that is under exploration right now, so we don't know yet the full potential of that data, but early exploration is showing potential access to medication information and lab tests that could be interested. So we're taking a current look at this data, which include over 1,000 records. Now, the RDCA-DAP platform, you will hear terms about it today. Really our platform for rare disease data aggregation at CPAF, platform dedicated to maximizing the utility of the data and the use of the data. Utility because we're really implementing, the team is implementing a specific format to maximize what you can do with the data, create innovation in the way you can discover data, analyze data, maximizing the use because there is a public interface that has been open for maybe close to 2 years now, where users can access the data and take a look at the metadata and then potentially make a request for patient-level data and analyze the data online. So we have some of the Friedreich ataxia data set available right now, including the aggregate, and some users are taking a look, including one of our panelist teams today, at the data on RDCA-DAP. So you can see here that is through the RDCA-DAP portal, just typing for Friedreich ataxia, you get a list of data sets, and in green the FA aggregate is available for request. And we are integrating, as I mentioned, the European data. The key angle here is that we needed to put that European data on RDCA-DAP because it's fully GDPR compliant and has been built so that we can also accommodate international data, which was really critical for us to be able to get that data shared to us to further the effort in Friedreich ataxia with that European natural history data. And you can see here, one of the key aspects is that there is a little more more security and privacy when you access data on RDCA-DAP. For the CODER database, it was a download of the data that you can get to use on your computer, but on RDCA-DAP, it's very controlled. You cannot download the data at the moment unless there is a specific request for that by contributors. So it's providing security on secured workspace with tooling so that you can analyze the data directly, and we're trying to build tools that you will hear about today to facilitate the data analytical directly on the platform. So this is an output that really shows, that I really like that graph. It really shows the extent of the data that we have, because this is data for the Friedreich ataxia rating scale, and the black dots are individual patients through the database. So you can see the tremendous amount of information that we have on one of the critical endpoints that is used to track Friedreich ataxia patients. So, we are programmed to RDCA-DAP, and our goal is to leverage the data and build solutions to accelerate drug development. I'm not going to go into details, but one of the ongoing work that we are doing right now internally is to develop disease progression modeling for Friedreich ataxia. We talked a bit about that in a session yesterday. We're trying to advance clinical trial simulation tool for the disease. That's the RDCA-DAP work. There is a tremendous amount of publications that came from the FAICD members, and we can send over publications. Typing David Lynch, Friedreich ataxia, or Christian Remy, to name two members of the FAICD, you see a lot of critical publications that were made throughout the years. On that note, I will introduce our panel today. So we have Jen Farmer, CEO for the Friedreich Ataxia Research Alliance, Shannon Rich from Rare Pharmaceutical, and we'll talk about the first approval, first drug approved for the disease that Rare had this year. And Shannon is Associate Director of Clinical Data Science at Rare Pharma. And Robert Chuck from OCP FDA. Robert is a clinical pharmacologist. I'll give you a chance to introduce yourself further, do a better job than me first. Jen, if you want to start, maybe tell us more about the efforts at FERA, and really what was the original intent for you to come to CPAF, to create that database, and where you wanted this effort to go.
Sure. Thanks, Alex. Hi, everyone. I'm Jen Farmer, and FERA is 25 years old this year. We were started by patient families and scientists, and as an organization, really focused on advancing research in Friedreich's ataxia. Most of our programs are centered around, funding basic translational clinical science grants, as well as, what some of the previous panelists talked about in terms of looking at that pre-competitive space in drug development and trying to put the tools in place for the community so that we can accelerate and further inform the drug development process. And so the inspiration for creating the FAICD came from Jane Larkindale, who's sitting in the back of the room, who was working with me at FERA a while back. At the time, we had been collecting natural history data for a little over 10 years. And we also had several failed clinical trials. And one of our previous panelists, talked about the importance of patient contribution of data and how hard it is for individuals participating in natural history studies and clinical trials to be contributing that data. And so we were really motivated to start this integrated database so that we were really leveraging every single contribution of patient data that was being put forward, whether it was natural history study or clinical trials. And we also knew we had to be smarter about how we were designing these trials, given the failures that we had had in the past. And so that was really, kind of those different things coming together simultaneously led us to the creation of the FAICD. And, we were really fortunate that a lot of the industry partners that we had worked with previously on those trials were really ready and willing to contribute the data from those trials to the database.
Thank you, Jen. And just to emphasize, it is a tremendous achievement to get so many studies, and it was actually pretty fast. So my day-to-day of trying to get data shared to our platform, it is quite an achievement to get that fast, and we're still building. Next, Shannon, if you want to maybe tell us more about your role at Reata, and then why it is important for industry to be able to access this aggregated database. And if you want to extend, we'll continue with you a little more to discuss specifically what you did on the FA data. But first, please tell us a bit more about your work.
Yeah, I'm Shannon Rich. I'm the Associate Director of Clinical Data Science at Reata Pharmaceuticals. I've been at Reata for 5 years, and I've had the opportunity to work on clinical development programs in Friedreich's ataxia, as well as other rare diseases. And I guess a little bit later I will share some more about my experience using the FAICD database. But first, to answer your question about the benefit of having an aggregated database, I think it's beneficial really throughout the drug development process. It can inform trial design, in terms of having that aggregated patient-level data that you can decide how you want to analyze. And so you can really answer very specific questions about a patient population that you're interested in. So getting information about disease progression and baseline characteristics, which can allow you to understand, the expected change from baseline and expected variability in a given endpoint, which can better inform the sample size of a trial. At the end stage, after the trial is completed, I think that having access to that data from other trials and natural history studies can allow you to really more deeply understand your own trial data, because you can examine your trial results compared to the other studies in the database.
Thank you, Shannon. Now, Robert, some question. Maybe tell us a bit more about your work within FDA, and also, maybe the angle from the regulator perspective and someone at OCP on being able to access this aggregated database, again, from the regulator perspective.
Thanks, Alex. And I think I inadvertently sent an older bio that's a little bit outdated. I'm currently the Deputy Director of the Division of Translational and Precision Medicine, which is still in the Office of Clinical Pharmacology at FDA. So I've been at the agency about 10 years, starting in the Genomics and Targeted Therapy group, which is a group that works on precision medicine approaches across different therapeutic areas. And a few years ago, we combined with a team working on rare disease drug development and really trying to use translational science approaches to advance drug development for rare diseases. So that's kind of our current goal, and one of our goals in trying to access the RDCA-DAP database. And really try to use it to figure out how we can come up with better approaches to develop drugs for these groups of patients. Why do I think it's important from a regulatory perspective? I think we're always, as others have said, we're working with a paucity of data in rare disease drug development. So we really need to take advantage of every piece of information we have about these patient populations in order to see if a drug is safe and effective for them. And that's where we can really use resources such as RDCA-DAP to combine data sets, look at biomarker development, design better clinical trials, and really make sure that we're doing the best we possibly can to give these drugs a chance to prove that they're safe and effective in the patient populations that they're being developed for.
Thank you, and apologies I scorched your title. Good job, Alex.
No, I think that was what I sent in.
All right. So yeah, Shannon, maybe back to you. Can you tell us more about really the Reata story? So again, the first approval for the disease arrived this year, which is like an amazing achievement for a patient population with a terrible unmet need. So, Jen, a comment first?
Yeah, can I just because of the prior session and the importance of patient engagement, and I loved the discussion about disruptors in the field and family members who become citizen scientists. That's actually where this story starts. And so I thought it was important to maybe just give you a little bit of that backdrop. There's a mom whose son was diagnosed with Friedreich's ataxia, and she was up every night reading every PubMed article she could find to understand more about the disease. And there had been a few publications looking at how the protein deficiency in Friedreich's ataxia leads to downstream metabolic and mitochondrial consequences. And she comes across this NRF2 pathway paper that has been published by some of the academic scientists in the community. And her husband was a chemist and supplying API to biotech and pharmaceutical companies. And so the next morning she says to him, NRF2, is that a druggable target? And he happened to know the answer and said, yeah. And she said, do you know any companies working on drugs that target NRF2? And it turns out he was working with Reata and helping supply them with API for some of their other products. And she picked up the phone and called Farah and talked to Ron Bartek and said, listen, this could be just some late night mom Googling and reading papers, but I just feel I have to share this with you. And Ron, of course, was all in and interested in what she had to say, and followed up a quick introduction by her husband to Reata with Ron, myself, and one of our lead clinician scientists, Dave Lynch, flying to Dallas and knocking on the door and saying, hey, let us in. We want to talk to you. We want to tell you about Friedreich's ataxia. And at the time, Reata was not at all interested in developing drugs for neurologic diseases. They were focused on kidney disease. And so we kind of really surprised them. This was kind of out of left field. But, I just wanted to share that as the backdrop with our prior session about, really engaging your community, the disruptors, and our role as advocacy organizations in facilitating that process and kind of bringing people together. That's where this story starts. And I'll turn it over to Shannon to also, talk about the clinical trial design and the work that we've done together since then. Thank you.
Yeah, thank you so much. So our trial in Friedreich's ataxia is called the MOXIE study. It was conducted in 3 different parts. So part 1 was a dose-ranging, placebo-controlled trial, which patients were dosed for 12 weeks. Part 2 was conducted in separate patients. It was a placebo-controlled trial as well as our pivotal study, and patients were dosed for 48 weeks. And then eligible patients from both the Part 1 and Part 2 studies could enroll in an open label extension phase. And so the extension began enrolling, I believe, in late 2018 and is still ongoing. And so we've collected many years of safety data as well as some efficacy data. And so in our Part 2 pivotal study, what we saw or what we observed was a significant improvement with our drug relative to placebo in the primary endpoint, which was change from baseline at week 48 in MFARS, which is the Modified Friedreich's Ataxia Rating Scale. And so with these positive results, we wanted to use the long-term efficacy data, MFARS, that we had collected in our extension phase to support these pivotal trial results. However, because it was an open label extension, we didn't have a control arm to use to kind of best interpret the extension results. So we requested the FAICD database, which had the FACOMS data, the natural history study, along with some other trial data, so that we could actually use that data to provide an external control to better understand our extension results. And so I guess more specifically, we used the natural history data and performed a post hoc propensity match analysis. With this technique, patients from our extension study were matched one-to-one with patients from the FACOMS study. And the matching was done based on sex and then other prognostic factors for disease progression that were available in both of the studies. And so this was baseline INFARS score, baseline GATE score, which is a question within the INFARS, age, and then age of onset. And so we came to these factors with discussions from FARA and Dave Lynch, who's been mentioned, and statisticians at FARA. And then I guess one other really important thing to mention is that within the FAICD database, there are different studies, but the FACOMS study was really the one that was beneficial to us because of its really large size. So it made matching one-to-one feasible. The FACOMS study is also the only one that collected the INFARS data over many years. And so obviously we needed, long-term data collection so that we could analyze our long-term results. They also were very robust in collection of demographic and baseline characteristic data, which is what we needed to be able to match those patients and really generate a comparable external control group. And so, all these factors really made the FACOMS data within the FAICD database the one we needed. And so after we matched the patients, we analyzed change from baseline in INFARS over 3 years in our extension patients and in the patients that they matched to within the FACOMS data set. And we observed an improvement in INFARS in extension patients after 3 years of treatment compared to the untreated matched FACOMS patients. And so I guess just in summary, having the FAICD database and the FACOMS data specifically really was beneficial in allowing us to interpret our open label extension results and in providing some support to our pivotal Part 2 study results.
Thank you, Shannon. Trying to see where to go next because I have 2 angles here. I'm going to start with the RDCA-DAP piece. So as I mentioned, we transferred some of the data that is already available. One of the reasons for that is as we want to continue the effort, there's a need for globalization in Rare. Really, if we want to get somewhere and push things forward, we really need our efforts to be international. And so RDCA-DAP was built in part for that reason in Europe to be able to enable that international aggregation of data. So some of that data is available. Robert, some members of your team are taking a look at the Friedreich ataxia data. Maybe can you give us a few highlights on, I know it's early, but some of the interest that maybe you see from your team's perspective on using the RDCA-DAP as a platform to take a look at rare disease data?
Yeah, certainly. So I think the case example is a great one of one potential use of this platform. I mean, it's highly impactful that they're using it for, actively using it for part of the regulatory approval process. That's very impressive, especially as early on as it is in the use of the database. So I'll also just say in full transparency, from the FDA's perspective, I was not involved in this review in any way, shape, or form. So kudos both to Riata and to the review team. I'm simply commenting here as a bit of a spectator. But I do see many other potential uses for this type of platform beyond just that direct application in the drug approval process. Our group is looking at it as more of a research tool. We're looking at different ways that we can use innovative approaches to advance drug development. One of those is better understanding the disease itself, and that's one big advantage I see of letting everybody be able to access this data. You have many different minds and many different people who can think about things in different ways and come up with ways to better understand a disease, and that can help us with things like biomarker development, pharmacodynamic biomarker development, looking at correlation between baseline factors and prognosis or outcomes in patients, really just helping us better understand what the, because we have such small numbers of patients, what we can expect to see in a clinical trial. And that, again, helps with that clinical trial design, helps us have additional endpoints, come up with ways to have confirmatory evidence of effectiveness. I think that's how the database was used in this case, was in regulatory speak. It's a way to bolster our substantial evidence package, is to have a piece of confirmatory evidence. One way to do that would be a natural history study, as was done in this case, compared to part of the trial. Other ways would be to have like pharmacodynamic biomarker effects can be used as confirmatory evidence, but we have to understand those biomarkers and how they typically behave in that disease. So by looking longitudinally across databases like this that have biomarker information, we can better understand whether something might be a good pharmacodynamic biomarker, whether that could be a piece of evidence we could use in a future submission. So we're really taking more of a research-based approach. It's still pretty early on, so we're kind of figuring out what's in the database, what we can use, what questions we can ask, but are looking at, kind of being able to better understand the disease as a whole and how the next drug development program might look like.
Great, tremendous. And like speaking on behalf of the team too, we really appreciate, all users, that are currently using RDCA-DAP getting back to us with feedback. We all strive to make the platform better, provide new tools and innovation so that we can really continue to improve what we already have, which is already great. So Jen, moving on now, I'm sure there were lots of tears of joy in the community, and then boom, the aftermath. This is not a cure for FA, and after years and years of work, it was a big relief, but now it's okay, moving on. Where do we go next? So maybe can you comment on what you see as ongoing challenges and maybe opportunities to continue the effort for your community?
Yeah, absolutely. There's the need. I think Pat said it beautifully. We've got to continue collecting this natural history data, right? We need to understand how, as treatments get approved, the natural history changes. We need to get new tools in place for the next generation of clinical trials. We need novel trial designs. And so we're actually kind of doubling down now on this initial investment and trying to grow it even further. And the European natural history study that Alex mentioned, we're actually combining that into a single natural history study, FA-COMS and EFACS coming together into a unified study, so that we can have more than 3,000 individuals with FA globally engaged in natural history studies, continuing to collect this data. Being able to support post-market registry studies, that's going to be really important to us as well. The conversations that have been happening here around pediatrics is also a high priority for us. The drug is approved currently in individuals 16 and older, and we need to start trials in individuals with FA younger and younger, and understand how best to do that. So, Pat came over to me and said, "How are you doing?" Because you thought it was hard to get to the approval. You get to the approval, and then it just really takes off, and then things get really crazy, and there's a whole lot more work to do. So, these kinds of efforts are ones that are, not just a 2 or 3 year investment. We started FA-COMS in 2003, and it is 20 years in now, and it has to be something that we can figure out how to sustain and grow so that we can continue to evolve our clinical trials and bring new treatments forward, and that's what we're thinking about. Great.
Maybe I'll relay the same question, Robert, if you want to start, maybe commenting now. We have a new baseline therapy. What's the impact, and how should we move forward with the future clinical trials?
Yeah, well, one thing I'll say is that often, once you have an effective therapy and you have patients on an effective background, it can become harder to actually demonstrate efficacy for the next drug unless they happen to be synergistic. So, thinking about, that means we need to do a better job with clinical trial design and all the things that we've been talking about. in terms of understanding the disease, designing the best trial, having the most effective endpoints that are going to be able to demonstrate an effect for the drug. And again, I'll kind of come back to what I've been saying. I think using it as a research tool to understand how we can improve all of those elements, improve that clinical trial design, discover biomarkers that can be used as pharmacodynamics, and able to show an effect there. And really just be able to prepare for what the trial is going to look like, because as everybody said, the environment changes once that drug is on board, and the natural history changes, and all the things that we knew aren't necessarily true moving forward. So, it's why it becomes even more important to understand the disease as well as we can.
Thanks. Jen, maybe a comment on that? I'm sure it's part of your consideration right now, continuing the data collection, like the baseline therapy.
Absolutely, doing all of that, and continuing to understand, how to develop better endpoints and biomarkers. At the same time, I think Michelle said it. You need to continue to evolve these processes, and so. At the same time, you want to keep collecting a lot of the same data so that you can compare over time, but you've got to keep iterating, and you have to keep innovating that for new endpoints and biomarkers. And, that is, something that I think we've done pretty well in our natural history study in terms of, evaluating potential endpoints and biomarkers in small pilot studies and thinking about looking at that data and deciding, okay, which ones do we bring in now that are part of the core set of assessments, continuing to look at the data we're capturing, getting rid of things that are not sensitive, that don't seem to be working well, and getting better and better tools.
Yeah, great. Yeah, and something like I didn't present, but really the FERA scientists did a tremendous job taking an overall look at 17 subunits of the scale. Is that 17? Yes, 17 subunits within the scale. Across the database, all these subunits are tracking patients over time, which subunit might work better for which category of patient at which stage. That's really a lot of effort. And it speaks also to how great it is to have a collaborative environment as we're building our trial simulation tool, the first core, as we paused the work for a few months. As we restarted, getting the feedback from EFACT and FERA on the effort, telling us, well, we don't really care about this is where you should focus on improving your models, because this is where we want to intervene. Seems obvious, but there's also a lot of conversation of, patients getting from ambulatory to non-ambulatory, the transition phase, how can we better find means to track and to measure that. There's, it's part of the challenges that we need to, and why we need to continue the work.
Maybe one other thing I'll say that's been, I think, a really good outcome of the FAICD and RDCA-DAP. FA is a small research community. It's relatively small. And we have one statistician that we work with as part of FA Comms. And being able to make these data sets available to the broader community, we've had more than 20, 30, I lost track, but I mean, we've shared this data with a lot of different companies and academic researchers and with the CPath team. And so we now have more and more people looking at this data, thinking about different ways to apply different statistical approaches to understand the data better, to help us with disease modeling, understanding the placebo effect that occurs in FA trials, and different approaches to clinical trial design for different therapeutic products that are in development. That's also been, I think, another important outcome, and one that we want to continue to facilitate is, having more and more people help us think about what ways we can leverage this data set for new trials and understanding the disease better, and it's been really important for that as well.
Okay. Maybe a final comment from Shannon on what's next for READA. Maybe what was missing, what you would want to see as the database continue to increase.
Yeah, I think as Jen mentioned, the drug is approved in patients 16 and older in the U.S. And so obviously, there's a focus to expand in the rest of the world, but also in children. And I guess just thinking about, kind of what's missing, or obviously, and you've been mentioning it as well, just continuing to collect more data. And you're doing it with EFACTS, but there are some differences in endpoints, and so getting that data that has more different endpoints will be more useful to other sponsors as well. I know just one challenge we faced while using the database was really just we were interested in concomitant medications, and it was collected in both studies, but there was some differences in the information that was collected, and we didn't know what coding dictionary was used, and so it made it challenging to really summarize those results across the two studies. We could get some useful information, but it was just hard to really directly compare them.
Mm-hmm. Great, thank you. And I think the HR piece, I think, might be something we need to continue to discuss. We talked in the past about maybe the lack of information on cardiac data. There's really still a lot to do. So I will stop there and start the Q&A. Maybe just take a minute to acknowledge the moms. And your story was a great listen to your mom story. And we had a group of data scientists and moms of rare disease patients yesterday. So thank you to the moms. And let's start the Q&A. No, Cecile, you're second. There's someone behind you.
Well, she can go because I was
French
actually going to defer to the woman who I jumped in front of last time. So go ahead.
French culture, we are sorry about that. No, I really want to thank the panel and this session about RDCA-DAP. So as I mentioned yesterday, I'm leading activities for CPAS in Europe, and I just want to really congratulate everybody, because everybody in Europe is so jealous about RDCA-DAP. Like, it's really a game changer, and it's really perceived with a lot of positivity and a lot of discussion about how to engage best and make it interoperable to accelerate really the development of global data set, to enrich data for disease modeling and DDTs, and I do hope that we can help you advance that project in a very efficient and timely way. So congratulations and thank you.
Now I have to come back with a question after all that praise. So, Shannon, you mentioned something that caught my attention about capturing the meds. And I'm wondering, actually, for the natural history data that you've been collecting, if you've thought about those factors beyond the clinical manifestations. A very good example of that is Erbitux, where they noticed some safety issues with patients who lived in North Carolina and Tennessee that was related to, eating barbecue, as we all do as Southerners. But like, that kind of information actually does guide the responsiveness to a therapy, but it's not something you'd capture in a clinical trial. So I'm wondering if you guys are thinking about that, or even for RDCA-DAP.
So Brianna and Farah are working together to launch a post-market study where we can capture more of that real-world evidence and longer-term safety in a broader population, and leveraging our Farah's clinical research network that does the natural history study to implement that post-market study. So I think we are thinking about that, where we know that the number of people who've been able to access the medication through the trials is relatively small. It's about 170 or so people, and now expanding that out to 4,000 individuals in the U.S. or there will be things to continue to learn, and thinking about how best to capture that. Yeah.
All right. Well, thanks everyone. Thanks to all the three of you for joining. It's always a treat to do a panel with Alex, I know. But thank you very much. We're heading into a fifth.